Okay, let's continue our discussion and have a detailed look at the gene expression or a continuation of gene expression. Now, when the genes are expressed, what happens is that it results in the production of proteins, and those proteins will usually have specific structure and even functions. When such a protein is made, it usually subsequently falls into a three-dimensional structure, of which that three-dimensional structure that results is the one that will enable the protein to have its function. So unless a protein has folded into its three-dimensional structure, usually it will not be having its functional capacity or its functional role. So it has to be immediately folded into its three-dimensional structure through the processing that occurs within the Golgi complex. Now, you'll find that some proteins are composed of a single polypeptide, while there are others which are made up of more than one polypeptide. In some cases, even up to 4 different polypeptides can be present within a protein molecules. Now, the messenger RNA molecules are not always messenger RNA molecules. They could be either tRNA molecules, they could either be ribosomal RNA molecules. Others could be the messenger RNA. Now, the relationship which exists between the gene sequence and the protein that is resulting from that gene sequence is of ultimate importance, and this will be especially with regard to the protein which is formed from the messenger RNA that has been formed from the gene which is present within the chromosome. Now, what determines the function of a particular protein once it has been formed within the cell? What determines the function of the protein is the gene sequence that codes for that protein, and after which that protein has been formed, of course the modifications which occurs to it within the Golgi complex will also be able to determine the function which that protein shall perform. Now, the information which is required to synthesize a particular protein, where will it be residing in? That information will usually be residing in the DNA, and we saw that the DNA is stored within the nucleus of the cell, especially with regard to eukaryotic cell. Now, if you consider the characteristics of an individual and even the characteristics of a cell, those characteristics will usually be depending on the type of proteins that are present within that individual. You saw in the first slide of our discussion of genetics, we saw, okay, in the second slide, not the first one, but the second slide, in the discussion of genetics, we saw a number of individuals there. One was a total African-looking person. Another was an African-looking person, but of maybe altered pigmentation. Another one was American-looking or European-looking. Another one was looking like a Chinese. Another one was looking like an Indian. All those people, their appearance is being determined by their genetic makeup, and therefore their proteins which are present within those individuals. Remember, I told you that the Indian person, their hair, the reason as to why it is straight and not folded or twisted like ours is because of the fact that their hair protein, as soon as the hair starts to form, for it doesn't what? It doesn't fold. But for the case of the African hair, once the hair props up out of the hair follicle, then automatically it starts to fold. And that is one key characteristic of the genes which influences their the characteristic features of an individual and how they appear, or how they actually they are made up with their physical characteristics. Now, if you consider the total collection of proteins within an organism, whether it's a human organism, whether it's a plant, whether it's a bacterium, some of the bacteria, they cause disease, and some other viruses, they also cause disease. Whatever the total protein collection in an organism, we shall call that collection as the proteome of the organism. Now, the functions of each of these proteins that are present within the organism, they usually be differing. Some of the present would be enzymes. Some of the enzymes would be involved in catalyzing metabolism within our digestive system. Other enzymes will be just responsible for catalyzing other functions within our cells. Then we also have other proteins. For example, if you consider tubulin, and tubulin is the one which is assembled into the large structures which are referred to as microtubules. And we know that the microtubules are part of the micro or tiny tubules and filaments which make up the internal structure of the cell, and therefore they help to stabilize the structure of the cells. In other cases, other proteins which have been expressed, they can be inserted into the cell membranes, and in being inserted into the cell membranes, they can facilitate the transportation of substances either into or out of the cells. They can also be inserted into the cell membrane, and instead of being involved with transportation of substances into and out of the cells, they can actually be involved as enzymes just present on the membrane of the cell. So in that case, they'll be performing different functions there. In other cases, some other proteins which have been expressed from the genes could function as biological motors, and in other cases, this protein could also be, for example, that protein myosin, and you know that myosin is so much involved with the muscle contraction processes. Now, if you consider the proteins which have been formed within any given cell, and then you consider the functions which they play, you'll get to know that the function of a protein is usually very, very important to the overall well-being of the cell. If a protein loses its function, then the overall well-being of the cell will also be in danger, because of the fact that each protein will be involved in performing specific functions within the cell. Now, let's consider how the genetic makeup of an individual can be linked to the physical characteristic of the individual. In this case, linking the genotype, which is the genetic makeup of the individual, to the phenotype, which is the physical characteristics of the individual. So what can you see in this picture? Are you able to determine what this picture is showing? Are these bands for maybe taking tea? Because there's a time that I asked students and they told me that these were bands for taking tea in breakfast. Okay, so these are not bands, these are actually red blood cells, and you can see at least 3 normally shaped red blood cells and one blood cell which is not normally shaped, it is altered in shape. Now this alteration in that particular red blood cell, not only itself but even other red blood cells which are altered in shape, results from an alteration in the genetic makeup of the individual, and subsequently an alteration in the structure of a certain protein present in the red blood cells, which is involved in the transportation of oxygen within our circulation. So which protein is that we're referring to, that when it is altered, it is also going to alter the shape of the red blood cells? Of course, that protein is, as you guessed it, hemoglobin molecule. Remember, hemoglobin is a protein, and of course, being a protein, it would be encoded by specific genes, and any alteration in the gene coding for hemoglobin will therefore subsequently result in an alteration in the structure of the hemoglobin, and in this case, that alteration in the structure of the hemoglobin will actually result in the alteration in the structure of the red blood cell, and that results in what we call as sickle cell anemia. I hope you have heard about sickle cell anemia, or sometimes you could have seen a patient who is suffering from sickle cell anemia. Is their health very good? Do they have normal health? Do they enjoy life and normal health the way we do, those who don't have sickle cell anemia? Your answer must have been a total no, because you have seen how their health deteriorates with time, and in some cases they even end up passing away at an early age because of that fact that their blood is not able to transport oxygen normally in their circulation as it's supposed to actually perform under normal circumstances when somebody doesn't have sickle cell anemia. So we shall see the intricate details of how sickle cell anemia results by seeing how the alteration in the gene coding for hemoglobin causes an alteration in the structure of hemoglobin itself, and subsequently how that altered hemoglobin structure results in an alteration in the shape of the red blood cells, which then gives us the name which we call as the sickle cell anemia. So we shall proceed with that in the coming video by looking at all that sequence of events which brings the alteration that results in sickle cell anemia in the individual. In case you need to understand more, check in the next video.